All right, hey, welcome. Welcome to the video. You've already seen two different videos today. One having to do with both parts of photosynthesis, the other part dealing with just the Calvin cycle. I hope you enjoyed that one with the Dr. Seuss looking characters. And I'm hoping that it was very insightful to help you understand what it is that we're doing. When I was making this video, this is my third attempt, by the way. It's late into the evening/night. And so I had this perfect YouTube video set up. I had a nicer camera, better camera angles. The camera is actually right here. I had it pointed back up at the screen. I don't know if you can see there's stuff that we're going to go over. Had it all, drew it out twice, and had problems with the camera both times that I was recording. And so then I didn't want to re-erase all of this again and redraw it because I'm making other videos as well. And so this is what we're going to get. This is the second time I drew it. I'll go through and explain. You can follow the mouse. If you don't have all of this written down, which none of you have all of it written down, you can always pause the video. You can always go back, rewind. If I said something that you missed, you can go back, rewind, watch it. But we're just going to go through, look at photosynthesis, talk about both the light dependent and the light independent video or reactions here, cycles. I've talked through this, 2 times now, and you think I would have it down exactly what I want to say, but now I'm a little jumbled. So I'm going to work through this again with you. Hopefully we can get this right, or I can get this right this time, and that you are learning and doing well, even while I'm not physically with you. And so with photosynthesis, if you were in the A-Day classes, we've covered the light dependent reactions, haven't touched the Calvin cycle, but B-Day, we started light dependent reactions. We didn't get to them very in depth, but I'm going to go back all through the light dependent. Then we're going to work our way into the light independent, and then we'll call it a day. So photosynthesis itself is going to, we're going to look at this equation of 6 CO2 plus 6 H2O. If you watched in the videos earlier, they've used this light energy, or they've written in light energy where this arrow is. That's because the light energy is what drives this reaction. Without the sunlight, you don't get the switch from the reactants to the products here. And so that gives us C6H12O6 plus 6O2, which is the oxygen. And so photosynthesis is going to start with the sun, and then we're going to be inside the chloroplast. We marked before, this outer boundary is the chloroplast. And then, let's see if I can zoom. It's better. All right, these dots that I've circled here are the chlorophyll, and these are going to be found inside this first protein complex called photosystem 2, which is abbreviated PS2 for short. And so these small dots are those light-absorbing pigments, chlorophyll. And so as we look through this, you actually have the entire, I went too far, electron transport chain right here, starting with photosystem 2, moving down, we have photosystem 1, a series of proteins that are going to hand off and use the electron's energy to move items through, and then ferredoxin, which is the final electron acceptor, which hands off the electrons for the electron transport chain. But before I get ahead of myself, let's go back, look at the light energy from the sun being absorbed by the pigment chlorophyll. And so light energy is absorbed, and then it's going to be transferred into these electrons. So you have these empty, we'll say empty. So we talked about this on the A-Day in terms of comparing it to a gas can. So if I zoom up, here we go. The electron itself is empty, much like if this gas can had no gas in it, you wouldn't be able to use it for much. And however, if we fill this gas tank up with gas, then we can then put that gas into something and it's worthwhile. the electrons in this case are going to operate a lot like this gas can. And so they're going to be a storage vessel for this light energy to be like held inside of. And then we're going to work our way of converting this from light energy into chemical energy. So the electron is then moved up this protein complex, photosystem 2, and the light energy is going to have this interaction here where it's going to fill it with energy. It's going to be indicated, we filled up, I colored it up so it looked more like that gas can. And I'll reference that analogy as we go through this. And then we're going to start our road trip. It's going to go down and work its way through the electron transport chain and eventually be handed off to these proteins, which are going to use the energy that we got back in photosystem 2 to help move these H plus across the membrane. Now, because we have only 2 H plus here, and then there are more below down here, this is going to be moving from an area of low concentration to an area of high concentration. And when you do that, that's called active transport. So the stuff that we learned last chapter is not going away. It's still here, still present. If you didn't learn it, then you need to get it done now. So the energy from the electrons used by the proteins to drive this fueling or to move these H plus across the membrane, because inside of this thylakoid, we are trying to build up a concentration of these protons that we can then let filter through this ATP synthase protein in another way. So the energy from the electrons is used to move H plus across through these proteins, which leaves our electron a little depleted on energy. But that's okay, because like any good road trip, you have to make a gas stop at some point. And so we're going to end up in photosystem number 1. PS1 is then going to recharge the electron, harnessing that same light energy as before using these pigments. You see them here as well. And so the electron gets refueled before it's eventually then handed off to ferredoxin, which is the final electron acceptor. Ferredoxin is then going to place this electron between NADP plus and H plus, the E negative, and there's actually a pair of electrons here. And so it cancels out both the positives with the negative, positive cancel out. And so that's how we then get NADPH, which ends up being this high energy electron carrier because it's carrying these electrons and harvesting energy that's going to be used as we move over to the Calvin cycle. We see that NADPH playing a key role down here. So the whole point of this is that we are now going to be taking energy from the sun, visible light, converting it into a transferable chemical energy that can then be stored in a long-term chemical energy form known as glucose. So how do we get the other things around here, and what purposes do these H plus serve? Well, we'll start with the H plus and then I'll work my way over here and work to water. But the H plus are going to fill the thylakoid membrane. And then as they fill, they're going to naturally move from an area of high concentration to an area of low concentration. And so when we move from high to low, that's passive transport. Passive transport with the assistance of a protein, remember, is then facilitated diffusion. Because this has got a plus next to it, it's considered a polar molecule. It has an electrical charge. And so H plus is going to be moving down through the ATP synthase, which the rapid filtration or movement of these H plus through this membrane is going to drive the energy needed to reattach this phosphate back to ADP, which we said was that low energy state molecule. And then now it's become ATP again, which is full of energy, and it's going to make its way to the Calvin cycle. And so the whole point of bringing these in is to fill this concentration and then help keep this steady flow moving out, producing lots of ATP, which this is energy that the cell can use. This energy gets used really quickly. And so this is a quick turnover. It's not a delayed effect. And so we need something longer term. And so that's why we're going to be working on making glucose over here. So we've talked about the transport chain. We've talked about the H plus. This process is called chemiosmosis. If you go back and look at the notes I gave you at first, it walks you step by step through these. And then at the very end, it talks about chemiosmosis, which is this process of producing ATP. And so, yeah, let me think through this. Okay, we've covered all that. So with water being split, what happens here is that once this electron that initially is filled up moves out, then we have to have another electron to come in and replace it, or else the process stops. This is why water is so important to plants, because without the water replacement here, the water being split and the electrons released, this process stops. We don't make NADPH, and then we don't do the Calvin cycle. So a water molecule, due to the energy here from this set of reactions in the electron transport chain, the light energy, water is then going to be zapped, split in half. The oxygen then leaves the cell. You see 1/2 O2 pairs with another O2 or another 1/2 O2. You make oxygen, which then leaves and goes into the atmosphere. Could also be used in cellular respiration in the mitochondria, but where we get the oxygen we breathe comes from this process. And so then the H plus, the H2 part of H2O, we've seen the O leave, and let's talk about the H2. The H plus then moves towards the synthase, which is continues to drive that chemiosmosis process. And then the electrons that were holding the hydrogen and the oxygens together, those are going to go into the photosystem and continue to resupply that steady trickle of electrons that move through the ETC nonstop. And so to look at that, if we look at the structure of a water molecule, then the oxygen itself is going to be separated from the two hydrogens down here, which are sharing covalent bonds. Remember how you have to have a certain number of electrons in each orbital, 2 in the first, 8 in the next one. And so because they're sharing electrons, they're going to be split. The oxygen goes, the hydrogen is removed, and then you're left with electrons that are then sent into the photosystem. And so that all happens from water passing by this photosystem too. And that's what drives some of these light reactions. I know I've gone through this quicker than when I was drawing this out, but as I mentioned earlier, if you have any questions, or maybe I didn't mention this, Tony, third time I've shot this. If you have any questions, you can put those down below in the comments, or if you're in the dorm, you can email me. I don't know how soon I will get back to you, but well, because I'm gonna be on vacation. But I'll try to at night at sometimes just kind of peek and see once you've watched this video, because I mean this will be on Wednesday or Thursday, so I'll be several days into my trip. It's not going to be waiting for you to watch this every night. But if you're confused, go back, put a comment in, and then I can either respond to your comment, I can email you a response, or when I get back, I'll just better know what to address coming into that Monday when I return. And so if you have questions, please do that. Put them down below so I can see them. If we look at this equation, then you see we've got carbon dioxide plus water gives us glucose and oxygen. We used water, we split the water, and that gave us the oxygen. And so now we need to move over into the Calvin cycle, and we're going to learn how we can input carbon dioxide and get glucose out of it. I'm just checking to make sure I didn't forget anything. I think we're good. This is just showing that this happens in the thylakoid and that the Calvin cycle happens in the stroma. All right. And this once again showing that all of this is happening in the chloroplast and in the plant cell. Some bacteria can do it too, but mostly we're just going to be looking at plant cells. So CO2 is going to enter into the stroma from the atmosphere. that comes from us breathing, from other processes that happen through human activities, natural processes, etc. So regardless of where it comes from the atmosphere, CO2, you've got your one carbon with your two oxygen attached. That's where the C and then the O2 comes in. And the CO2 is going to be met up by rubisco, which is this enzyme that is going to exist and function here in the stroma. What rubisco is going to do, and this was the Dr. Seuss looking character from the TED Talk video, is he's going to match CO2 with this 5-carbon molecule, RUBP. And then when he does that, we get this 6-carbon compound that then gets split into a 3-carbon compound. So we go from having 6-carbon molecules to 12 3-carbon molecules. If you're confused there why we split that, that's just part of this process of carbon fixation, which is step 1. We didn't lose any carbons. If you take 6 and multiply that times 6, you get 36. Take 12 times 3, still 36. We haven't lost anything. We've just structurally changed what's happening because CO2 is an inorganic compound. And in order to get to glucose, we have to switch from inorganic to organic. And so that's what this first step is. It's called carbon fixation. We are fixing the carbons into an organic molecule. And so then these 3 PGA exist. And so I kept this so that we would see where all these are. And so really it would just be one of these groups of 3 would be a 3 PGA. And there would technically then be 12 of these floating all in this area at this time. But for the simplicity of keeping up with the original 6 of them, I just kept it in this form. So that's why it looks the way it does. So as we move into step 2, which is reduction, ATP that was made down here at chemiosmosis is going to come and energize. We're stuffing and packing in energy into these molecules. We're giving up our electrons, and even we're going to give up H+ that then is attached. So we have both a chemical and physical structural change. And so with a physical structural change, we're not the same 3 PGA we were up here. And so now we're a molecule of what's called G3P. And so the difference between these two is that we have a slight structural change because we've added the hydrogen and the electrons, and we've stuffed it full of energy from ATP. And so high energy molecules that are then going to be moved out to make these sugars. And so we had 12 3-PGA up here. We now have 12 G3P here. 12 minus 2, because we're going to lose 2 of these 3-carbon molecules, because 3 and 3 equals 6. And that's what glucose is a 6-carbon molecule. If we go back up and look, C6 tells us that. And so we have 12, we lose 2 to make this sugar, this glucose. Glucose then leaves the cell, or not the cell, but the chloroplast. And it's going to go out usually probably towards the mitochondria so that it can be used for cellular respiration, or it'll be stored. And eventually one of you will eat it and your body will break it down. But once we lose 2, we then go from 12 G3P to 10 G3P. All right. And so at this point, we need, in order to keep this cycle going, to convert back into RUBP, which is step number 3, regeneration. So we'll use ATP energy from chemiosmosis. You can see how to spell that there. We'll use it from this ATP from chemiosmosis. ADP will go back and get recharged. But this is going to convert this from 10 3-carbon molecules, 30 carbons in total, to 6 5-carbon molecules. Still 30 carbons. We've just gone from groups of 3 to now we're adding on to groups of 5. And so from structure of 3 to a structure of 5, and then rubisco can work some magic and attach to the next round of CO2 that come into the cell. So how does this part work? Everybody always gets confused here. Most people are okay with carbon fixation. Yeah, we go from 6 to 3 and, it's fixed or whatever to be organic. We're shoving energy in here, and people kind of understand that. You lose some to make glucose, because that's the point. You input your 6 carbon, dioxide to get this 1 molecule of glucose. And so now the conversion back, the regeneration, we're going to be moving from essentially 10 G3P to 6 RUBP. So we're going from 3 to 5 with the help of ATP. So the way that I structured this was that if you take a group of 3, or if you take 3 of these groups of 3s, that gives you 9 carbons. Split that 9 in half, break off 5. You now have 1 of the 6 RUBP that you need to make. We're then going to add some more. I color coded where they came from on this side. So you add 2 more sets of G3P. We have 10 total carbons here. Split those 10 in half, you get 5 and 5. There's the second and third RUBP made. So now we've made 3 of the 6 that we need to make. Put a question mark here because once we do that, in this instance, we split this and had some left over. Here we don't have any left over. Good news is we used 1, 2, 3, 4, 5 groups. And if there was 10 of them and we've used 5, that means there's 5 remaining. So we just repeat the process. Take 1, 2, and 3 groups. That gives you 9 carbons. Split off 5. There's your 4th RUBP that you need to make. Add in 2 more groups to give 6 more. That should add 4 plus 6 gives you 10. Split in the middle to get 5 on each side. And there's RUBP number 5 and RUBP number 6. So that's how you convert from 10 G3P to 6 RUBP. ATP is the one driving all of this and giving the energy needed to make the structural changes and rearranging. Why does this happen? I don't know. Just good design, probably. But it's needed to help sustain this conversion of molecules in this, addition of CO2. And so you're making the end products that then go to the beginning, become the starting products, and it keeps this cycle going forever and ever and ever and ever until the plant dies. And so that is the Calvin cycle. We do all of this to make 1 glucose that can then be sent to the mitochondria to extract a numerous amount of ATP for all kinds of different energy needs and production in the cell. And so if you have any questions over this, go ahead and write them down. I know that somebody has at least a question, at least one person does. And so if you don't, I'm willing to go through this again when I get back. That's fine. But make sure you can always go back and watch again. Go rewatch those other videos if you get the chance to tonight or in your free time sometime if you need help understanding, because once you see this and you go back and watch those videos, do that enough times, it'll start to click and make sense to you. I promise it will. And so that is the Calvin cycle, also known as the light independent reactions, and they happen in the stroma, which is that fluid area out here. Both of these cycles work together. And so they are interdependent on each other. The light reactions take energy from the sun, store it up, and then the Calvin cycle uses that stored up energy then to pack it several, energies into it, because glucose can hold that energy for longer than ATP or NADPH. And then that moves to get extracted elsewhere in the cell, the mitochondria. And so these work together. Affects us mostly because it provides the food that we need. This glucose provides energy in the food that we need. We get this glucose by eating, whether you eat plants or not directly, something that you eat ate the plant to get that energy. And as a primary producer, it supplied energy to the food web to eat. So the animals you ate or whatever had to get their energy from somewhere. And so this is where it all starts. The oxygen that we breathe comes from this process. So it is important to understand it. If anything, it helps you just appreciate what's happening. But that is all I got for you on this. So If you need to, go back, give it a rewatch. Yeah, or pause it. You can finish writing in. I can leave this up. Maybe try to make it zoom out a little bit more. Only thing you're missing now is chemiosmosis down at the bottom. But yeah, go in, draw this, finish it, have this in place. It'll help you with your worksheets you're going to do. I hope you have your book. That'll also help you. But until I get back, make sure if you have questions to go and to drop your questions down in the comments. Make sure you like the video and smash that subscribe button and turn on the little bell for notifications. Not that I'm going to be posting any more of these, but who knows, I am trying to do some videos from our trip. So if you want to watch those, you can go watch those too. And I hope that you have a great rest of the day. See ya.